Okay, so I think we will start the session right now. So, could someone please let me know whether the screen that I shared is visible and whether my voice is audible or not? Okay, I see the chat. Perfect. Okay, thanks. Okay, so again, I welcome you all to the live tutorial session for the contents of week 9 for the course Bioinformatics Algorithms and Applications, instructed by Professor Michael Bromia from IIT Madras. So, as usual, in this live tutorial session, we will just go over some of the concepts that Professor Bromia talked about and introduced in his slides for the week 9. And also, we'll just solve some of the numeric questions that are related to sort of the topics that he introduced and similar questions basically which came in some of the earlier iteration of the courses as well, right? Okay, so just for a quick recap, in the past few classes, we talked about calculating some numeric distances, for example, using the coordinates from PTB files. Then we learned about several statistical parameters, for example, root mean square deviation, as well as radius of gyration, which can lend us insight into the structural deviation or the basically protein folding state via analyzing the compactness. Then we talked about hydrophobicity, several concepts related to that, for example, long range order stabilization center, conservation score, and ultimately like combining all of that to basically calculate certain free energy changes. We also did those things. And then we talked about what sort of changes, depending on what sort of changes, depending on the surrounding environment, the certain mutations can be being forward basically. So we talked about charge changes, then space filling, then static hindrance due to certain mutations, depending upon the size of the residue that is being mutated and the size of the new one, whether there is enough space for that enlarged sort of side chain to accommodate or not, basically depending on the surrounding environment, that can impact the stability or conversely the destability of the mutation, basically the mutated structures, right? We calculated some of the binding free energy calculations, and based on that, we would try to infer the packing of certain residues before it was mutated. So we also did these kind of calculations, and then we talked about, for example, depending on the surrounding environment, what sort of uh, mutations would bring forward what sort of stability, flexibility, hydrophobicity, and so on, right? So in the classes of week 9, Professor Bromia introduced several sort of concepts related to the binding free energies, for example, binding of protein ligand complex, and then certain terms related to the binding that we always have to analyze when we talk about certain binding like protein ligand. It could be protein, it could be protein nucleic acid, and so on. And then in the same vein, he also discussed about that we discussed about in the earlier classes some of the concepts regarding protein folding. So he also introduced some concepts regarding calculation of free energies based on the different states of the folding, and then using different kinetic experiments, for example, we can try to infer some of that. Uh, so some of these concepts are discussed. So in this particular class, again, I will go over, for example, excuse me, some of the concepts, and then we'll try to solve some questions basically. So if we try to talk about, let's say, the very first question that I put in my slide, which we'll also look at like one example of using some softwares, right? So let's say you have a protein ligand complex. Now, how can you get a protein ligand complex? Of course, you can search for your interested protein, and let's say if your interested ligand is already co-crystallized with that protein, so probably some research group have submitted the 3D structure for the protein ligand complex, like in the PDB database. So you can get a particular protein ligand complex in the PDB database. Alternatively, what you can do, you can, for example, if you have a certain small molecule you are interested in trying to find out whether this molecule could be a effective binding partner for a protein at a certain binding site, right? So what you can do, you can go forward with molecular docking. So that is one approach that we will probably discuss in some of the later classes. So molecular docking is something that we can do, right? There are various softwares available freely where you can perform molecular docking. For example, you can use Autodock Vina. And if you are interested in protein docking, for example, a lot of web servers are available. For example, Haddock, HDoc. So they basically can allow you to dock different molecules, whether both of them could be bimacromolecules or one could be small molecule, another would be bimacromolecule, right? Now, when you talk about a protein ligand complex, so you can get the complex via docking also. So there are several terms that we always try to investigate, or basically these terms are always used in relation to the investigation of the binding between those protein ligands, right? So basically, if I ask, for example, what does those terms indicate essentially, right? So the first one is binding site, second one is binding mode, the third one would be binding free energy. So can anybody, let's say, answer maybe in the chat or unmute yourself and try to say like basically what these terms indicate? So what would the term binding site mean for a protein ligand complex? Then what is the difference between the binding site and binding mode? And then what would be a good way to basically good way to talk about the binding free energy? Like what sort of energy are you talking about here? Just okay, so I see answer in the chat. Binding site is basically active site, allostatic site. Yeah, you are talking about the different types of binding site that could be present in some proteins, right? So binding site is essentially, if you talk about in general terms, so it is basically the region or site of the protein where the ligand binds. So that means where the interacting amino acids are present in the protein, right? So there should be, you can imagine there should be some sort of cavity present there, which will allow the ligand to basically move into the cavity, kind of like bind to that cavity. And conversely, the binding site, when you describe binding site, of course, you have to describe in terms of which amino acid residues are present in that binding site. And then after that, you can proceed forward with an analysis that, okay, so I am trying to investigate, let's say, the interaction energy between these particular residues which are present at the binding site with our ligands, right? So essentially, the region where the ligand binds within the protein, and as I see in the chat, so it can be, of course, active site. So active site essentially means that the catalytic region, basically, of a protein, of course, since it's the catalytic site, so there should be some sort of physiological partner, let's say physiological binding partner that could be present in that, which in physiological condition which attaches to that particular site and then does some signaling, right? So that would be the active site. So we can also try to, let's say, block that active site in our target protein so that we can prevent these sort of interactions. If these interactions are unwanted in certain diseased conditions, then you can try to inhibit that catalytic site or active site, right? Conversely, as we talked about allostatic site, so allostatic site is some, basically these are some sites where certain molecules can bind, but instead of showing its immediate effect in the particular allostatic site, it has some long-reaching effects, kind of, right? So where the binding site, where the allostatic site is present, basically where the ligand is binding in that allostatic site, it shows, let's say, certain conformational changes in a distant region in that protein. So we can say that, okay, that is probably an allostatic site, right? It could also have implications in drug discovery for as well. So if you have certain allostatic sites where some ligands could bind, and then that could basically lead to certain conformational changes, or conversely, it could lead to restriction of certain conformational changes that we want, basically based on our needs. So in that way, we can basically design allostatic drugs against certain protein molecules, right? So those are basically binding site. Binding mode, as I see in the chat, so it is covalent, non-covalent. You can, yeah, these are basically the different categories of binding mode, we can say. But in general terms, again, the binding mode will essentially indicate the geometry of the ligand at the binding site. Now, when you dock certain small molecules against a protein, uh, you will see that in the docking results, more often than not, the software will give you different conformations that can be possible of that ligand within that binding site, right? So the difference between those conformations will be uh, they will have certain different orientations or at certain different chains or around certain bonds, let's say. So there will be some difference in angles, torsions, and so on, which will basically mean that it is like slightly different conformations, right? So these type of different conformational deviations, or like what sort of particular geometry or conformation that the ligand is attaining while it is present at the binding site. So that will be considered the binding mode of this particular protein ligand interaction, right? So the geometry of the ligand or the particular conformation of the ligand at the binding site, and you will see in docking software basically corresponding to each binding mode. So there will be certain differences between these different binding modes. So they will provide, let's say, different docking scores. They will calculate different docking scores. You will see that, right? So binding mode is also important in terms of investigating the protein ligand binding. And the final one is, of course, binding free energy. We talk a lot about free energy changes. In terms of binding, it means that the free energy of complexation, right? So the free energy of complexation means, let's say initially you have, we also discussed it in some of our earlier classes also. So initially, if you have a protein, initially if you have a ligand, they are unbound. So they will have certain energies, right? Now after binding, so in the protein ligand bound complex, the final energy that will also be something else, right? So if you basically subtract the final energy, which is the complexes energy, minus the initial energy, total initial energy, which will be the individual contributions by the receptor and also by the ligand, as you can see here. So if you subtract the final minus initial, you will get a delta G. So delta G is change in G or change in energy, right? So that will be the binding free energy, what we are talking about here. So we also know that it can be calculated using this formula, delta G equals to del H minus T delta S. So delta H here is talking about the enthalpy change upon complexation. And then this T delta S is the contribution for the entropy or the correction that is required for the entropy part. So with total, if you combine all of those, we can get the delta G value. So you will notice in certain cases that the delta H is also further can be further decomposed into different kind of components. For example, there will be energies corresponding to van der Waals interactions. There will be energies corresponding to electrostatic interactions. There will be energies corresponding to solvation, polar solvation energy, non-polar solvation energy, and so on. That in all total will give you the binding enthalpy. Combining with entropy, you will get the binding free energy, right? So these are basically the three terms that are always used and investigated when we try to talk about protein ligand binding. Conversely, protein binding, it is also the same thing. So any type of binding that we talk about, we have to investigate these sort of terms, right? I think in one of the earlier classes, I talked about some algorithms to calculate this binding free energy from emitted trajectories. So one of the popular approach is called MMPBSA, molecular mechanics, Poisson-Boltzmann surface area. Conversely, another one is called MMGBSA, so molecular mechanics generalized Born surface area. So this generalized Born part, it is kind of an approximation of the Poisson-Boltzmann approach. So it is, since it is an approximation of that, so it is slightly, let's say, less accurate, slightly less accurate. But even MMPBSA, that does not give you the absolute binding free energy, so it also has certain limitations. The best calculations for binding free energies, which also take a lot of computational power, will be something like thermodynamic integration or free energy perturbation. So these are some of the techniques that can be used to calculate absolute binding free energy, though they are quite computationally expensive. So you see that basically you see the approaches like MMPBSA, MMGBSA the most, because they sort of have a balance between the computational cost and the time consuming part basically, and also decent accuracy, right? Okay. So in the next slide, what I have just put is a 3D view of a particular ligand bound inside the binding pocket of a protein. We'll see that the protein is in ribbon structure. And of course, the small molecule or the ligand is visualized inside, right? We can see this yellow colored structure here. So this particular region where the ligand is binding will be called the binding site, right? And the binding site, of course, is composed of many different amino acid residues. We can see some of them are highlighted here, right? Like isoleucine 226, methionine 256, phenylalanine 401, etc. So these will be participating in some sort of interactions with the ligand molecule, right? And conversely, the type of geometry that the ligand is attaining, or basically the particular conformation that the ligand is attaining, that will be our binding mode, right? So that is sort of represented visually in this particular slide. So we'll look at another tool that can let us visualize the binding mode or the binding site just in a bit, right? So we'll use Chimera for let's say general 3D visualization. And if you want some sort of 2D ligand protein interaction diagram or interaction map, we can use another software called LeakPlot, which we'll just see in a bit. Okay. So yeah, here I basically had one example. I think we also saw some similar example in some of our earlier classes. So this is basically calculation from a MMP-based binding free energy approach, right? Again, you can see the complex minus receptor minus ligand part that is done for two different calculations. So one is for the enthalpy calculation, which is sort of the upper box here, so this upper calculations, and then another one was the entropy calculations. Combining both of them, you can get the binding free energy. Okay. So as I said, the energy component for the enthalpy was divided into different components. For example, van der Waals, electrostatic, polar, non-polar, etc. Conversely, the entropy part also has some components: translational entropy, rotational, vibrational, etc. You can get the total entropy change corresponding the contribution of the entropy change and combine that with enthalpy to get the total binding free energy. Right? So just quick question: can somebody say, for example, if this upper calculation is of the enthalpy and then this bottom calculation is of entropy, what would be the delta G value here? Can somebody just maybe calculate it out real quick? So the upper part is the calculation for the enthalpy, and then bottom part is calculation for the entropy. So what will be the
minus 13.4?
Yeah, minus 13 or minus 13, right? So what you have to do is basically you have to do delta H minus T delta S. So if this is part of the T delta S calculation, then you have to kind of do minus of minus 17.8647. So it will be essentially minus 31.4 plus 17.8647. It will come around minus 13, right? You can see also the associated standard deviation as well as the standard error of the mean. So these are here because when we calculate the energies using some molecular dynamics trajectory means basically a collection of snapshots, right? So there are thousands of snapshots, lakhs of snapshots that are present in the trajectory, and we basically pick out certain region of that MD trajectory, and we use that for the calculations, right? So that means, let's say if we use 2000 snapshots in total for calculation of the energies, then the software or the program, what it will do is that it will calculate each of the individual snapshots energy components, right? And then ultimately it will average it out. So because there is, let's say, this 2000 snapshots, so along with the average value, we will also get the standard deviation value and the standard error of the mean value. Okay. Okay, that's fine. You can also calculate the propagated error for the in the delta G binding also. You can do that by basically using square root of this delta total or delta H total square, the standard error of that square, and then plus this delta S total, that contribution square, and ultimately you just do a square root at the end. So you can do the propagated error calculation also this way.
Excuse me, from where did you get this table? Matlab, with some software or from where?
Yeah, so this was basically a data that we got from a molecular dynamics trajectory. So for MD simulation, you know that there are several softwares available. One is Amber, which we have used in this case. Another one is Gromax, which is also quite popular. So we have used Amber software to simulate the trajectory, simulate the certain systems, the protein ligand systems, and then the calculation was again done using one part of the Amber suite, basically, which is called CPP Traj. It's a calculation module, basically. So this is basically an in-house calculation. So we have uh, run some simulations of certain protein ligands, and then we have found this data. So that's just for demonstration.
So,
I think
is there some software or supposedly if I have to do this analysis, so what am I supposed to do? Matlab, if I
uh,
first thing is I have a novel protein. So let us suppose
yeah.
I have a novel protein, so I don't know which ligand it is going to bind. So is there any database or somewhere where I can get some tentative idea?
Yes, yes. So usually the protocols for the novel drug discovery, when you do not have, let's say you do not know already what kind of ligands are binding to it. So usually what we do, you can take one approach. If you have already known ligands, then you can create some QSR models. But alternatively, if you know the binding pocket of the protein, binding region of the protein, and if you have a 3D structure of that protein, you can first go for a molecular docking approach. So molecular docking is basically you trying to fit one ligand into the binding pocket of a protein, and then you're trying to check for different conformations where the energy will be uh, the lowest, essentially, right? Because that will indicate stable binding. So an extension of this molecular docking approach will be virtual screening. Now, virtual screening is basically when you have, let's say, thousands of or lakhs of molecule. So you need to have basically 3D structures of those. You can just do a docking for all of those molecules against your protein. So that approach is called virtual screening. So there are different softwares available for molecular docking, etc. You can try in Autodoc, Autodoc Vina. I think Autodoc Vina also allows virtual screening of large libraries. There are other softwares commercially available, for example, Schrodinger, MolSoft, etc., which will allow you to basically do a virtual screening with a large library against your target protein. And the software output, what it will give, it will of course give you the docking score or the binding energy score in terms of the docking protocol, and then it will also give you the binding mode. Basically, it will give you the protein ligand bound structure, 3D coordinates of that. Uh, Now, since docking is kind of like a static kind of approach, so it only provides you one snapshot. The protein is rigid, only the ligand is moving. Basically, it is trying to identify the perfect geometry or the conformation of the ligand that can fit inside the protein, which will give you the lowest energy, right? The protein is rigid there. So that's why the accuracy of docking protocols are quite, low, right? Of course. So what you can do next is the output from the molecular docking software or the virtual screenings, whatever you have got, the protein ligand bound structures, you can simulate that through a molecular dynamics protocol. So simulation essentially means that you will sort of virtually simulate an environment, let's say solvated environment where certain salt concentration corresponding to the physiological environment is also present, and then the protein ligand are sort of present inside the bound structure of the protein ligand are present inside its solvated box, and then through the integration of Newtonian formulas for motion, right? Uh, and then the different potentials that can arise from that. So those are basically described in different force field. So you can use those to basically identify what the complex will behave like in a dynamic system. So that is what molecular dynamic simulation is basically is. And after you have obtained a collection of snapshots for a trajectory, you can go for these sort of MMP-based calculations using the trajectories. So usually what sort of usually basically the software that you are using for the molecular dynamics simulations, like I said, Amber, Chromax, etc. So these softwares will also have tools for calculating the binding free energies. Okay, so you can probably look those algorithms up. So I think MMPBSA, MMGBSA are widely available in and incorporated into these different programs. So you can probably use those algorithms or approaches to calculate the binding free energy, basically. So firstly, you have to do a virtual screening. Then with the structure of the virtual screening, you will also get a corresponding docking score. So if you think that it is worth proceeding forward with that docked structure or the dock pose, then you just take the 3D coordinate and you simulate it in a dynamic system corresponding to, let's say, you are trying to see what sort of behavior it will show in a physiological kind of environment, right? So it is not exactly a complicated environment because we cannot uh, easily replicate the physiological system, but we can replicate the salt concentration, we can replicate the solvated environment, etc. The simple things, right? And you can proceed for a simulation and then analyze the trajectory basically. Yeah.
Okay. And one more thing, you mentioned about virtual screening. So this, whatever softwares are available, so I have to do one-on-one, means like so I have with my protein target,
Yeah.
I have to do one-on-one, or this software automatically gives me a list of some, let's say 10 protein ligand complexes, so making my work easier.
Yeah. So usually the virtual screening softwares, they have an automated process basically. So you do not need to do one docking after one docking after one docking manually. But if you, let's say, prepare your systems carefully at the beginning and then you provide all of the structures that you want to dock, basically screen, right? So it could be thousands or lakhs of compounds. So the software will basically just continue doing the docking one after another. So you don't have to manually interfere in between, but ultimately we'll get the output for the best docked structures, basically best docking.
Okay, thank you.
Yeah, yeah. So we'll look at some examples of docking in, I think, in some future classes also. But yeah, so alongside the binding basically concepts, Professor Gomiha also went over different sort of terms and concepts related to protein folding, unfolding, and so on, right? So one of the questions, let's say, I think some of these were introduced in Professor Gomiha's slides. So he talked about certain amino acid residues that were more prevalent in slow folding proteins, whereas he also showed us certain amino acid residues which are more prevalent in fast folding proteins. So if this question is asking, let's say, which of these residues are predominantly occurring in slow folding proteins? So could you answer this in the chat maybe? So the slow folding versus fast folding, they do have certain uh, basically preference, you can say, in terms of the types of residue that are present there. Okay. Yeah, I think I see one answer B in the chat. So of course, as you might imagine looking at these options, so it basically depends on the polarity or conversely the hydrophobicity in this question. Okay. Now, what Professor Gomi has showed us, so he also introduced us basically different tables which detailed some calculations regarding the composition of certain residues in slow or fast folding proteins. Now, here the answer would have been alanine and leucine, and we will see that the basically the non-polar sort of residues, for example, here we have alanine, leucine, and so on. We will see in slow folding proteins, they have higher basically composition value as corresponding to the fast folding proteins. You can see here from 7.5 to 9.7. So of course that it does not mean that these residues are not present in fast folding proteins, but it's just that they have slightly higher preference for them in slow folding proteins, right? So 7.5 to 9.7 we can see in cystine's case, we can see fast folding proteins have only 0.4 while slow has 1.2, so that is a 3 times increase. Uh, Glycine is also 7.6 to 9.5. Leucine is also 8.4 to 9.2. Conversely, what we see is that polar residues such as asparagine, glutamine, lysine, serine, so these have preference in fast folding proteins. So these fast folding proteins case, if you see the composition values corresponding to the slow folding proteins, there is definitely an increase, right? So that is what Professor Grom had kind of introduced. And he kind of reasoned it in a way that the hydrophobic cores, when they're formed, so they basically involve long range interactions. Okay. Now, these long range hydrophobic interactions are basically, they basically take time to form or stabilize kind of thing. So this kind of slows down the folding processes. So that is why the hydrophobic residues are basically hydrophobic residue dominant proteins, so they will kind of be a slow folding protein. And conversely, he also talked about like the different residue pairs, for example, alanine, 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 glycine, 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 tryptophan, leucine, methionine, glycine, etc., etc. So these sort of different pairs are more prominent in kind of slow folding proteins. Conversely, of course, we can see that the polar residues are more prevalent in fast folding proteins. Okay, so they have a more sort of dominant short and medium range contact interactions. So these short and medium range contact interactions, of course, take a smaller time to form. So because of that, the folding is kind of fast in a way. Okay. Now this folding of slow versus fast, we'll also see it kind of also depends on the dominance of secondary structures. Based on that, we also classified our proteins in one of our earlier classes, right? Alpha dominant, beta dominant, etc. So we'll see that also in some of the next slides. But in this case, the answer was alanine and leucine. The hydrophobic ones, they are more kind of predominantly present in slow folding proteins. They have slightly higher prevalence there. Okay. Now we are talking about protein folding. So of course, if we want to investigate protein foldings through experimental, through computational approaches and so on, so we need to have an idea about, okay, what type of time scale we are talking about when where basically we expect to see unfolded to folded state, folded transition, right? So if we just simulate our protein using from an unfolded state, let's say we want to see the different intermediate stages and so on, and we ultimately want to reach the folded state. So what sort of time frame do we have to simulate our system for, right? So what time frame is required for a protein from an unfolded state to reach a native 3D conformation? So it is a quite long range of time. It's not that it is a small range. So the range is quite big, but in terms of the time scale, what sort of values we are talking about here? So to see basically changes in the folds of certain proteins, usually we are talking about uh, time scale of over microseconds. Okay. Now, in this particular case, the answer would have been a microsecond to millisecond time scale. So, microsecond time scale is basically when you can start, to see certain changes in the fold. Maybe it also depends, of course, on the size of the protein. If it is a small protein, then of course it will take less time to fold. If it's a larger protein, then of course the entire fold to its native conformation, that will take much longer time, right? So overall, if you talk about from unfolded to folded state, the time scale could be in microsecond to millisecond times, right? So you can imagine now if you want to basically investigate through a computational approach. So running millisecond long simulations is not that easy. So it takes huge amount of resources. So it's quite hard and quite challenging in that way, right? Okay. So that was one question. So that's why if you had like sort of a unfolded structures and you want to identify what sort of 3D structures will be the native one, you would have to run the simulation or something for a very long time to predict the native conformation. Okay, so very challenging stuff. Yeah, I see one hand in the chat.
Sure, is simulating millisecond for millisecond is more harder than simulating for microseconds.
Yeah. So what I'm talking about simulating here. So when we use molecular dynamics simulations, uh, we basically use a time step. So time step basically means after how much time point the new sort of coordinates will be calculated using the potential and all. And the time step that we use in molecular dynamics simulation is 2 femtoseconds, generally in that range, right? So you can imagine that if we want to simulate a nanosecond time scale, so we have to use that, let's say 2 femtosecond will be the first calculation. After that, the next calculation will give us 4 femtosecond. The next calculation will give us 6 femtosecond coordinate. The next will give us 8 femtosecond and so on, right? So this way, the entire calculations will proceed one after another. And ultimately, at some point after a lot of calculations, we will reach nanosecond time scale. Similarly, whatever we have reached at the nanosecond time scale, if you want to extend that up to the microsecond time scale, we have to do 1,000 times more calculations, right? Because of course, nano to micro, let's say it's 1,000 times more steps will take to reach that time scale. So you can imagine that we have to do that many calculations. And similarly, from micro to millisecond, again, we will need to do a lot more calculations, right? Again, 1,000 times more calculation from millisecond time scale, right? So basically, because the individual calculation steps are in terms of femtoseconds, so we have to do each calculation femtosecond wise, right? 1 femtosecond, 2 femtosecond, then 4 femtosecond, then 6 femtosecond, and so on. And of course, if you have a much larger time scale in milliseconds, so it will need a lot of calculations to reach there. So that is why it will take more time and also more resources, because you need to have strong computational power to achieve this sort of calculations, right? In a weaker GPU, it will take maybe months, months after months. It will take to reach these sort of time scales. Yeah.
So, but the calculations, calculations, the program is inbuilt for calculations.
Yeah. The program is,
Already.
yes. The program is built for calculations, but for each calculations that it does, it will only do in this femtosecond time steps, because that is very much required if you want to accurately calculate the sort of next set of coordinates that you want to calculate, right? So whenever you are talking about simulating at a larger time scale, you have to perform a lot more calculations. So it will take more time. Of course, let's say if you have a GPU, you are running 100 nanosecond simulations in that. So it will take relatively a much less number of calculations. So you can, let's say you are getting your result or output after, let's say 1 week, right? In 100 nanosecond simulations. If you extend that up to 100 microseconds, then you are talking about months, right? Because it will take obviously longer time to reach that time scale. It will have to do all of the calculations in the intermediate steps, right? And then if we're talking about millisecond, then that is again a vastly larger time scale than microsecond, right? So it will take more time basically. So that is what we are talking about in terms of computational cost, because computational cost is basically all of your computational power will be occupied for that much longer amount of time to calculate this. So these are challenging to basically achieve essentially.
Okay, sir. And sir, is there any example of the fastest folding protein and the slowest folding protein like that?
Uh,
In literature?
yeah, so on the top of my head, I cannot think of an example, but if you have, let's say, smaller protein, I'd say that would probably be fast folding, because there will be less intricate kind of folding in the core in between, right? If you have a much larger protein, then you can imagine when it folds, basically it has to account for all of the possible conformations for the residues that are present in that, right? So again, larger proteins might take larger time to fold. At least when I'm talking about this, I'm talking about, let's say, in terms of simulating these sort of things. So of course, if you want to predict folding of a large protein, then it will take longer time to simulate. I'm just talking in that sense, but perfect examples of these fast and slow, I do not have immediately in the mind. I'll have to look it up actually.
Okay, sir.
Yeah. Okay. Uh, so yeah. Now, so for example, here we have another question. So it is asking basically energy-based approach to identify the binding region in protein-protein complexes. So which sort of interactions are used to calculate or to identify the binding region or the binding site? So we also talked about earlier the binding free energies and so on, right? So what were some of the constituting components in those binding free energy calculations? So that will kind of give you the answer.
Sir, are we talking about forces here, like van der Waals and
Yeah.
electros?
Yes, exactly. So it will be based on the van der Waals and electrostatic interactions and so on, right? So these are basically used to identify the binding site in the energy-based approach, right? Now, since we are talking about binding sites, so I think we should kind of look at one example maybe. Just a moment. Yeah. So I hope the screen is visible. Uh, in earlier classes, we talked about one visualization program which is called Chimera or UCSF Chimera, right? So it is made by the group, the research group at University of California, San Francisco. Okay. Now we can use the same. So let's say if you have a protein ligand complex, you have docked the uh, ligand onto the protein, or you have just taken one PDB structure where a protein is co-crystallized with the ligand, and you are interested in finding out, okay, which binding or which protein or which amino acid residues are interacting with this ligand, right? Then what you can do, one approach is that you can open up the structures, the 3D structures in something like Chimera. So it will give you the visualization, right? Let's say here we have one protein. Now for better visualization, I can just change some of the colorings and backgrounds and so on. So I will just go to the color option. And here, let's say I will change the background to white. I will change the representation where this preset maybe. Let's hide all of the protein residues. And we'll focus on the ligand first of all, right? So we can just select the ligand here. And give it some color, right? So here we can see one particular ligand is bound to the binding region, right? If we are interested to see, okay, where is this particular binding cavity where the ligand is inserted into, we can try to view a surface representation maybe, right? So it will calculate the surfaces. And if we do the surface representation, we'll see that, okay, so the ligand is sort of bound inside this cavity. Now, of course, if you are interested in drug designing against a target, you have to particularly study the binding pocket carefully, because it's not that a ligand can just go bind anywhere. you have to particularly target that ligand inside a particular binding pocket of interest, right? So you are interested in preventing some sort of physiological interaction or mediating some sort of allostatic regulations. So you have to identify your binding site or binding pocket accordingly. That's one thing. But let's say I just turn off the surface view. I can just surface hide here. Now let's say I want to see, okay, so which basically binding site residues are involved in the interaction, right? So one approach is that we can just select the ligand. Okay, we can do it in this way, or we can go from here and then select inside structure. We can select ligand also. So one particular selection mode that Chimera allows is something called zone selection. So what we can do is that after selecting our particular ligand in this Chimera visualizer, we can select around 5 angstrom distance of that ligand. So whatever amino acid residues are present in that range, it will be visible. Okay, so we can do this zone selection, and you will see some part of this ribbon is highlighted, right? We can go to the sequence viewer, and you can clearly identify which amino acid residues are present within a 5 angstrom distance range from the ligand. Now, this is kind of a standard sort of distance range. You are expecting the binding residues to be inside of this 5 angstrom region, right? So that is one kind of standardized value that we use sometimes. But here, essentially, the highlighted residues can be viewed in the sequence viewer, and you will get, okay, which exact residues are present at the binding site, right? So
Are the green ones or the yellow ones?
yeah,
Which one are the?
no, the green ones. So here the green ones are basically the highlighted residue. So let's say if I want to highlight this region, so I can just use that in the sequence viewer. You can see that it was highlighted in a sort of a light green or lime green color, right? So these are the highlighted residues. So let's say it will make sense when we just look at side by side, right? So let's say I want to select the ligand. So you can select the structure, you can select the ligand, we can go to the zone selection. We can select a 5 angstrom zone, and then it will see that this lime green highlight is sort of present, right? Hopefully that is visible. Okay. So after that, you can, maybe if you are interested in visualizing the side chain orientations, then you can try to go to the actions, and then you can show the atoms and bonds, right? Like that. So here I think some visualization problems are here. So that is why we are seeing some sort of fragmented stuff. But in general, so it is a good approach to, like, in broadly identify the binding site basically, right? Which residues are present here. You might need to input some of this information in certain calculations for your MD trajectory, let's see if you're analyzing those. So these residual informations could be useful essentially, right? So that is one way to sort of overview the binding region and what amino acid residues are present. But let's say if you have a protein ligand structure and you are interested to identify, so which residues are sort of involved in what type of interactions and so on, like hydrophobic interactions, hydrogen bonding, and so on, right? You have a dock structure. Although most, let's say, a lot of docking softwares or virtual screening softwares will have some tools to view the 2D protein ligand interaction diagram, but let's say you don't have that available right now. So if you have a protein ligand complex structure, you can use one particular tool or software, which is this one. So I hope you can see. It is called LeakPlot Plus. Okay. So you can go to the software's page. You can, let's say, get some academic license. So it will ask your first name, last name, department, and then your institute, and so on. And then it will probably allow you to download the current versions of the software. Okay. So you can do that. You can install that. So we have installed in our Linux system. So let's say I want to use this leafplot software to visualize the complex that I just showed in 3D visualizer, right? So I think I have to do something like this. Yeah, so the leafplot terminal is open now. We can just go to the file option and then we can go to the open option, open the PDB file, and here we can basically, you can put a PDB code, but since I have the structure available in my local system, so I'll just browse and we can see this complex.pdb is present here. So I'll just open that up. So it is saying it is a complex, right? Complex.pdb. The PDB code is complex. I think that is because of the naming here. And inside that we have one ligand, small molecule, which is named UNK1. So it is basically an unknown compound. Let's say it was at the initial stage. So that's why the naming for the particular residue was UNK. The ligand was named UNK. Now we can press run just here. And interestingly, we'll see a lot of information was pasted in our terminal. But alongside that, this sort of visualization or interaction diagram was available to us, right? Of course, here you can see certain residues are inside these sort of radiating circular or sort of radiating arc. You can say these red arcs, right? So these are basically the hydrophobic residues. I think you can go to the edit options and then somewhere you should find the colored options, I guess, right? So here those different things are basically mentioned, what kind of color codes are indicating what. And then you will see the green residues. They are basically talking about the hydrogen bond forming residues according to the color code. We can see the distance of these bondings also mentioned here. So these different residues that are in the vicinity, which are particularly involved in the interactions, will be shown here. I think you might not be able to see all of the atoms of the residues or the ligand atoms conversely. So you can go to the edit option and then you can basically modify the size parameters to uh, clearly like visualize, right? So if I just want to change the size of the atom names, let's say to a higher size like 0.3 to 0.5, I think that will be clear enough. And let's say the residue names, I change the size to 0.8 or something like that. And then if I just press OK, you will see that the naming is a bit clearer, I guess, right? So let's do this 0.6. It should be a lot clearer, right? You can again rearrange the text that are just adjacent to the image. So it also allows you to do that so that there is no overlap when you change the sizes and so on, right? And it will overall like give you a an interesting view of the protein ligand interaction diagram. So you'll be able to identify which residues are involved in the interacting, which sort of atoms are being involved in the interactions, right? So these sort of things you can get from here, from this 2D diagram. Okay. So that is sort of
So
the link plot
just a
stuff.
minute, just
Yeah.
this circular ones are from proteins. This
Yes.
glutamate 172 or serine 150. These are amino acids of the protein, the complex.
Yeah. Yeah.
And this is the ligand.
Yes. Yes. So the ligand structure is this one, the atomic sort of structure that is present inside. That is the ligand structure. These red circular ones, these are the proteins that are involved in hydrophobic interactions. According to the color code, you can see different color options and so on from this color selection table. And then we also see some residues are basically indicated in this green color. So these are the residues that are involved in hydrogen bond formation. And you can see which atoms are taking part in the hydrogen bond formation with the ligand also. That is also mentioned in this dotted, let's say, green line, right? So those are the amino acid residues, and inside structure, that one is the ligand. So if you want to investigate the 2D protein ligand interactions and which amino acids are particularly involved in interactions, so you can use this software, the LinkPlot Plus software. It also has one suite for running protein interactions also, that is called DimPlot. So it is a part of this program only. So you can take the academic license for this one, and then so you can download the software and then you can execute the program for your particular complex. Okay, this will just allow you to identify the residues, and it is also widely used in different literature also. So the publications also, a lot of time they will ask you if you have some studies regarding protein ligand interactions. So they will ask you, okay, you provide some interaction diagrams, right, along with your calculations and so on. So you have to basically make a figure from these sort of softwares, and then you have to provide that to the journal sometimes, right? So it is useful. So this was one of the programs that I was interested in talking about. Uh, so these sort of interaction diagrams are also available inside different commercially available basically virtual screening softwares like your Schrodinger, your MallSoft, and so on. So you can use those as well. But conversely, this is one free version, so it is quite helpful. Quite helpful, right? Okay. So I'll just stop presenting from here, and then we can move with the further slides. Okay. So in the next question, what is basically asking is that say you have investigated the binding regions, right, and whatever residues are involved in binding. Let's say it's a protein-protein binding, so a large interface between two binding partners are present, and you want to see, okay, so we have this binding interface, different residues are interacting together. So which residues have more predominance in the binding regions, particularly in our protein, right? Protein interaction. So here a similar question is sort of being asked here. So we are calculating for arginine, and the total number of arginine in the protein is 2414, right? And 366 of this arginine are involved in binding sites. They are present at the interface, binding interface, right? So what is the binding propensity of arginine? So the propensity will indicate that uh, basically how much involved arginine is in binding, right? So basically kind of like that. So you can calculate it in terms of percentage maybe. So can you try to do the calculation and share in the chat? Okay, so I see 15%. So it's a kind of simple numeric question, right? So we have certain arginines that are involved in the binding interface. We have the total number of arginine. If you want to calculate the propensity, so we can just uh, take the number of binding bound arginine or arginine involved in binding. We can divide that by the number of total arginine. We will get a percentage, right? So in this case, 366 by 2414. We will get 0.1516. So if you convert that to percentage, so it will be 15.16%, right? So very simple numeric calculations. You can do it in this way. So these sort of calculations are they might come in your assignments and so on. Okay. Okay. So okay, here we have one question. So it is asking that in which of the following scenarios a computer-aided drug design program will not work? It is talking about the availability of different components that are useful in the drug design protocols. So firstly, it is asking we have known ligand protein structure, then known ligand and unknown protein structure, the known protein and unknown ligand structure, or both of them are unknown. So in which of these cases the drug design protocol will not work? Yeah, so I see one answer D in the chat. Of course, it is about availability, right? So you need both the protein structure, you need both the ligand structure for a drug design protocol, because ultimately in these protocols you will have to calculate some sort of binding free energies, right? So you'll need the 3D coordinates of your target protein and of your ligand. So just availability of the 2D sketch of the ligand or the sequence of the protein is not enough in this case, right? So you need the 3D coordinates of that, and both have to be available to kind of for a computational drug design protocol to work. So in the particular case of A, B, and C, we have some availability. We don't have the others. So maybe we can try to predict the other ones. But in case if you have none of this available, then it will be hard to do some drug design protocols, right? So it will be answer D in this case. Okay. Now, one of the basically widely used pharmacological parameters, or that is used in drug discovery projects and so on. So to evaluate your compounds, right, small molecular compounds. So one of that is Lipinski's rule of 5. Okay. So Lipinski's rule of 5, there are certain criteria that a drug molecule has to fulfill for it to be considered as a potential candidate. And one of them is log P. So do you know what should be the value of log P? The maximum value of log P allowed as per the criteria of Lipinski's rule. So what is the maximum log P value allowed as per Lipinski's rule for it to be considered a potential candidate, drug candidate, right? So it is called the rule of 5 because in all of the criteria, the threshold value or the cutoff value that are proposed are multiples of 5. Okay. In case of log P, the rule states that it cannot exceed a log P value of 5. The drug molecule should not exceed a log P value of 5, right? If we take a broad look at the Lipinski's rule, we will see certain things, right? So here, first of all, the content of the rule is the molecular weight has to be less than or equal to 500 Dalton for the small molecule for it to be effective. The log P value has to be less than equal to 5, so it cannot exceed 5. The hydrogen bond acceptor numbers for that molecule should be less than or equal to 10. The hydrogen bond donor atoms for that molecule should be less than equal to 5. And this particular table that I took, so it is from one web server. We'll just look at it in a bit. So it is one web server which is called Admit Lab. Okay. So it is a server where you can basically provide this mile notation of a small molecule, and it will be able to predict using its trained model what sort of admit behavior that this molecule is likely to exhibit, right? So just take a look at it in a bit. For this particular program or server, so the decision that they take based on the Lipinski's rule calculations is that if it has less than 2 violations for these 4 basically criteria, so that will be considered as pass, right? So it will be excellent.
Excuse me, this is
Yeah.
rule of 5. Rule
Yes.
of 5, right?
Yeah.
But there are only 4 parameters, no?
Yeah. So the Lipinski's rule of 5 has its name not because it has 5 criteria, but because all of the parameter thresholds that they have given are multiple of 5. So that is how it basically gets its name. Okay. So the 5 essentially means that all of these values here, the threshold values or the cutoff values, they are multiples of 5. And in total, it has only 4 criteria basically. So in this particular case, the admit lab server, so they decide that, okay, if it is less than 2 violations in total, so that is a pass, means we can proceed forward with that molecule. It is excellent in green, right? If it is had greater than or equal to 2, so that will mean poor. So less than 2, of course, means 1 or 0, right? So if it has no violations or only 1 violation, then it is fine. If it is showing 2 or more violations, then it is not good, it is poor. And in this particular case, the question was about log p, right? So what is log p? So the n octanol partition coefficient, that is called KOW. So it is also symbolized as p. So it is basically denoting the partitioning of substances between n octanol and water. So in a mix of n octanol and water, what amount of the particular molecule will be present in the n octanol part or versus the water part. So you have to take a sort of a fraction of that, and then you can convert it to log form, like the question here itself asks, right? So the P is an alternate symbol for KOW, and it is essentially called the n octanol water partition ratio. Okay. Now it is basically an indicator of the substance relationship between lipophilicity and hydrophilicity, water solubility, fat solubility, and water solubility. Okay. So if the KOW value of like if it is exceeding 1, so that means that higher solubility in fat-like solvents, because you are dividing the solubility in fat by the solubility in water, kind of, right? So if it is more than 1, then it will be more soluble in fat-like solvents like in octanol. It is less than 1, then it will be more soluble in water, right? And you can take basically the log value of that. And the Lipinski's rule is saying that if it is not exceeding 5, then it is fine. It's not a violation in that case, right? So Lipinski's rule is, it was introduced quite a long time ago. And currently, a lot of FDA-approved drugs, you will see that they do not follow all the criteria of the Lipinski's rule, right? So there are violations definitely. For example, in the molecular weight department, if it is saying that it has to be less than equal to 500 Dalton, you will see larger drugs have been approved by FDA, right? 700, maybe 750 Daltons, that molecular weight drug are also available. So Lipinski's rule, while it was a useful criteria, a useful sort of reference a long time in the field of drug discovery, nowadays researchers kind of take it not as a hard and fast rule, basically, right? So they have to also do different other analysis to validate the possibility of this drug candidate. And we definitely will see that a lot of drug candidates that are violating a lot of Lipinski's rule, they are being clinically approved, basically, right? So that's good. So it is not a hard and fast rule. It is just a reference to use in evaluation of the molecules, right? Now the particular program that I talked about here was Admit Lab. So if I go to the server of the Admit Lab, wait a second. So this is kind of the interface that you will get for the Admit Lab's latest version, I think 3.0, right? Here you can go to the help tab, and then you can click on the documentation part. So it will open up the documentations for the Admit Lab. And here you can see basically different components of Admit Lab, and then the admit evaluation, the screening portion, right? The newly added basically values and components and so on, right? So that will be the program description. Under the program description tab, you have a lot of different, for example, tabs as well. So you can take a look at that. But in the services tab, you can go to admit evaluation. And here you can basically put a smile string, a smile notation for a small molecule. You can submit that, and it will give you a prediction for the admit properties of that molecule. So for example, here we have an example. So if you tap the example tab, it will just automatically generate its a smile. So this is their example sort of structure. If you submit it, it will take some time to run maybe, and then it will provide you with all of the predicted values for these different parameters, right? So for a simpler visualization, they have this kind of notation. So green means it's good, uh, yellow means it's moderate, red means it's not good, right? So they have this simple sort of visualization as well. Accompanying that, they have certain values, which are probably different probability values based on certain calculations, right? So on and so forth. You can investigate all of these parameters. For example, it says overall physiochemical properties, then absorption, distribution, metabolism, expression, and toxicity as well. So if you see more greens, that means it is more good. But if you have to basically thoroughly analyze each and every one of these properties to see whether it could be suitable for your purpose, basically your particular interest, where you are trying to develop a drug against, right? So that's one server. There are other servers, for example, AdmitSAR, Swiss AdMe, Protox for toxicity, and so on. So usually it is recommended to not use just one server, but to validate the result using different servers and see whether they have the same predictions or not, right? Because you are relying on the trained models of that particular group. So it is uh, usually good to validate against different type of models, right? So different servers in this case. So that was one server for admit prediction. You can look through the documentation. You will find the interpretation for the data for Lipinski's rule and so on here. Okay. Now in the folding concept, the concept of protein folding. So Professor Bromiha went over something called a chevron plot, right? So what does a chevron plot, what is it useful for? So what does it basically indicate? Or what is it useful for? So can someone answer in the chat maybe?
Sir, in X-ray crystallography, we use
So in this case, the chevron plots are particularly used while investigating the folding kinetics. Okay, so the kinetics of protein folding, uh, that is where the chevron plots come into place. Now, in the left hand side picture here, so if you talk about just a simple kinetics kind of plot for just simple demonstration or visualization, so it is talking about an experimental protein folding landscape for a two-state protein with no intermediate. So what are these two steps? One, we can see here this D, uh, that is basically the denatured state or the unfolded state. Another thing we can see here, something called N. So this is the native state. Okay. So it is saying that it is a two-state protein folding experiment uh, or folding landscape with only one unfolded or denatured state and one native state or folded state. We'll see some delta G or energy values, energy differences between those two states. And we'll see in terms of energy values, of course, the native state has lower energy. That means it is more stable, right? So that's why uh, the folding will occur towards the native state, because of course, if it is more stable than the uh, if it is more stable, then the protein will try to achieve that folded state, right? So it is also given here, kind of, right? So one thing that's mentioned here, something called this TS, which if you look up in the figure explanation, it is talking about the transition states. Now, what are the transition states? So in this particular one, the figure heading is talking about that it is a two-state protein folding kinetics or folding landscape with no intermediates. So are transition states not intermediates? What could be the difference between these two maybe? So you can get that answer if you look to the left-hand side plot. So it is basically a general sort of reaction energy plot kind of, right? You will see that we start with our starting materials. So the starting materials will react with one another. Ultimately, we will end up with this PR or products, right? But between going from these starting materials and the products, we will see that different states are present in this energy sort of landscape, right? We will see these TS1 and TS2 are termed as transition states, while a relatively lower energy state from this TS1 and TS2, so that is termed as the intermediate state or I, right? So the transition states are basically highly energetic states, so high energy states where a lot of bond formations are breaking are happening, and due to that, these states are very, very unstable. So you will probably not be able to capture a conformation in a particular transition state because a lot of bond formations and breakings are happening, and these are very short-lived states, right? High energy, short-lived states. So those are transition states. Intermediate states are some sort of relatively more stable state which we get in between the starting and the end point, basically, right? So you will most likely be able to capture certain conformations. If we talk about, let's say, folding, you will be able to capture certain folded conformations which are not fully folded, maybe half folded, half unfolded, something like that, right? But which do not have a very high instability. Of course, it is always moving forward towards a more folded state, right, in a standard environment. But you will be able to capture those sort of relatively stable intermediate conditions, right, intermediate stages. So those are basically the intermediate steps where it is not as highly energetic state as the transition states, and you are able to capture some conformations in those states as well. Okay, so that is basically where chevron plots kind of come into play. So this is the kinetics of protein folding. And if you talk about the chevron plots, okay, so firstly, I have used the references as mentioned here. So you can go through these links and then look at it in a bit more detail. But if we talk about the chevron plots in particular, so it will look something like this. Okay. Now, what do we have here? So on the x-axis, we have some sort of compound uh, which has units in molar, right? So in this case, it is guanidinium chloride. So it is a high denaturant, very strong denaturant. Okay. And on the y-axis is presumably some sort of folding parameter, right? So if we take a look at the examples or the explanations for this one, so we'll see that the chevron plot basically includes the denaturant concentration versus the folded, let's say, uh, part of a protein. So some parameters regarding that in a dotted sort of form, but it does not basically include only one specific kinetic experiment data, but for a lot of different separate kinetic experiments data, which are of course inclusive of unfolding experiments and folding experiments. So all of these data are present inside this plot. Now, of course, if you are trying to do a unfolding kinetic experiment, so what you have to do, so this is the red line here. What you have to do is you start with a certain folded state, right? And then you progressively add more and more denaturants into the solution. So if you add more denaturants like this guanidinium chloride here into the solution, so what will it do? It will, of course, do the unfolding of the protein, right? It will induce unfolding in that protein. So you can sort of get the different percentage of folded versus unfolded structures in each point, right? You can make a graph of that and basically relate that with the particular concentration of the denaturant that you are using. And here this red red line that are being drawn over these sort of dark basically data points in this way. and also extrapolated to a much lower concentration of the denaturant. So this red line, that is indicating the unfolding experiment. Okay. So you have to monitor the changes with something like CD, circular dichroism, or fluorescence, right? In folding experiments, it is kind of a reverse. So it is indicated by this blue line here, and whatever black dots are coming inside this blue line, they are part of the folding experiment. So in this case, you start with an unfolded structure. So that means you start with a, let's say, solution where the denaturant concentration is basically high, right? So start with that, and what you do is you progressively dilute the particular solution. So you can say that after certain dilutions, of course, denaturant concentration will become lower. So conversely, if it becomes like low enough, so the protein will start to fold, basically. So that is the way to do the folding experiments. Here denoted by the blue dashed lines. And again, the procedure is kind of mentioned here. So folding is slower in the presence of denaturant than in pure buffer, of course. Unfolding is faster with higher denaturant concentration. And this sort of gives you the characteristic V kind of plot in the chevron plot, right? Now, here the folding or unfolding rates, or the K value, the logarithm of that is directly proportional to the denaturant concentration, so which are depicted in these dashed lines. Now you can extrapolate the data, as I said here. So you can extrapolate these dotted lines, and you can try to measure, okay, what was the folding rate when there was no denaturant concentration, and what was the unfolding rate when there was no denaturant present, right? So at zero denaturant concentration, you can extrapolate the data for the folding as well as the unfolding rates, and you can use that to basically get this Kf and Ku, and you can use that to calculate a free energy change, basically. So here we will see Kf of H2O. So H2O means the rate at water. So at water means no denaturant is present there. And Ku is again same, the unfolding rate in a water system. So no denaturant is present there. So these are obtained by extrapolation. And you can use this formula. So del G equals to RT ln Kf by Ku to get the free energy change from the unfolded to the folded states, right? So this sort of way you can get some energy values calculations, right? If you have sort of different kind of interaction partners, so it could be multi-domain protein, isolated domain protein, and so on. So we'll see that depending on these sort of constructs, the folding rate or unfolding rate can be slightly different. For example, in the first example, what we have here, so it is talking about the isolated domains data versus a multi-domain constructs data. And here it is saying that a domain folds at the same rate in a multi-domain context, but unfolds more slowly. So here we see that the folding is at the same rate, but the unfolding for the multi-domain protein is slower. And this is because of the domain-domain interactions and so on, right? Similarly, in another example, we will see. So it is again another multi-domain construct. So I guess I should have talked about the particular examples here. So here it is saying that if the neighboring domain of this domain-domain construct, so if that offers favorable interactions in the solely in the native state, so it is not in the unfolded state, then the kinetic barrier to unfolding is elevated while the kinetic barrier to folding is unaffected, and that is why you are getting this sort of plot. On the second hand, the next slide basically, we saw that the neighboring domain offers favorable interactions in both native as well as the transition states, and these interactions are compatible. Then the kinetic barrier to unfolding is unaltered while the kinetic barrier to folding is slightly reduced. So again, we are seeing this sort of pattern in this plot as well, right? So again, another example was that when the neighboring domain has favorable interactions in both native and transition state with stronger interaction in native state, then the unfolding barrier was elevated while the folding barrier was reduced. So basically, you are getting in essence different sort of patterns in the chevron plot based on the type of interaction or favorability between different domains in a multi-domain construct protein. So you can use these sort of plots to analyze those type of behavior as well, right? So that is kind of the point here. So you can go to the particular links that mentioned here and probably look into it in a bit more detail. But for the next question, what is being asked basically, that it is talking about contact order. Okay. So contact order for protein A was 8.7 and contact order of protein B was 5.7. Now, based on the contact order, can we identify which of these proteins will fold faster, or conversely, which of these proteins will fold slower? So can we try to analyze or reason through it? So does the folding rates depend on the contact order? Is there any available data for that? So I see one answer B in the chat. Yeah, in this case, the answer would have been answer B. Now, what is contact order particularly? If you look at the formula, so if I put the entire slide here, so in this contact order, we can calculate it using this delta Sij. So it is basically the sequence separation between contacting residues. Now, contacting residues, we know that it means in the 3D space, provided a certain cutoff distance, which two residues basically are lying within this distance, right? So that means that those residues are forming contacts in 3D space. So in this particular case, let's say the threshold was at 6 angstrom, and this delta Sij will be the sequence separation between the contacting residues. Ultimately, you have to do a summation of that, and you have to divide that by L times N. So what is L? L is basically the total number of residues. N is basically the total number of contacts. So that will give you an idea of the contact order. And in this slide, which was introduced by Professor Michael Brumiha in his lectures, right, we'll see something called the relative contact order in percentage. Corresponding to that, the LNK, or the rate of folding, right? We will see sort of this kind of negative slopes here. So it basically indicates that whenever we have more long range interactions, because remember that the contact order is sort of kind of indicating of this type of interactions, right? So if we have more long range interactions, then you will notice that the contact order is high. Now, why would that be? Because of course, since in contact order we are calculating the sequence separation between contacting residues. So if there is a high sequence separation, ergo in the case of long range interactions, so that will give you a higher value, right? So that is why the contact order will be high, and corresponding to that high contact order, the folding will be slow. In case of short range interactions, of course, that means the sequence separation will be much lower, right? So that will indicate that the contact order will be very less, because we have to calculate the sequence separation for that, right? So contact order in this case will be much lower value, and conversely, the folding will be faster, right? So sort of an inverse relationship between the contact order or long range interaction with folding is observed here, right? Fast or slow folding is observed here. Now again, in similar vein, if we ask that what will be the correlation between long range order and protein folding rate? So earlier we talked about contact order. So based on the sequence separation of contacting residues. Now we are talking about long range order. So again, I think we can imagine that since in earlier cases also the long range interactions basically were kind of correlated with the contact order values, right? High long range interactions, that means high sequence separation between the contacting residues, higher the contact order, right? So there seems to be some positive correlation between the long range order and the contact order. So in the same vein, since we saw inverse relation between the long range order and the protein folding rates, so we can say in the same way that in this particular case, we will also see negative correlation display, inverse correlation kind of thing, right? Again, what was long range order? So long range order was, in the case of contact order, we calculated the sequence separation explicitly. In the case of long range order, if a cutoff, let's say, if we have a cutoff value of Nij, so if this cutoff value or of this sequence separation, if it is more than 12, for example, so if the value of Nij is more than the cutoff value of 12, so we'll just put a 1 there. Otherwise, we'll put a 0 there, and ultimately we'll do a summation, right? In this particular case, Ij will mean that these two residues are forming contact, right? So if they have a long range kind of a contact, so we'll put 1 here. Lower than 12, we'll put just 0 there. So the 3D space interaction cutoff is kind of considered as 8 angstrom here, and then the n is the total number of residues in a protein. You can calculate using this formula, the long range order. And here, for example, Professor Gomiha in this slide, he introduced so the correlation coefficient versus the residue limit for long range contact. So here, for example, in this formula, we used the residue limit of 12, right? But for different residue limits that you set, then if you correlate that with the long range contacts and so on, right? So you will see ultimately that these sort of like 10, 12, 13, these residues are showing sort of the best correlation here. And we can see the correlation have basically negative value. So that means inversely correlated or negative correlated, you can see. Okay. So both long range order and contact order will be inversely correlated to the protein folding rate, right? Now, since both will be inversely correlated, so which could provide a higher value of this inverse correlation or negative correlation in the protein folding rate, whether it will be contact order or long range order? So in this case, our answer would have been actually long range order. Okay. We can see in the plot here. So for different categories of proteins like all alpha, all beta, or mixed proteins, Professor Gomiha introduced in this slide these calculations for the folding rate against the contact order and against the long range orders. And we see a correlation value, negative correlation value of higher in the case of the long range order versus the LNK plots. So here we see basically the absolute value is higher. That means it is more negative here. So it is better able to represent the correlations, negative correlations between the with the folding rate, basically, right? ln of the folding rate. In this case, the long range order would be the answer. Okay. Now, again, I mean, similar kind of question is given here. So we saw that contact order was negatively correlated with protein folding rate. We saw long range order was negatively correlated with protein folding rate. So then what is the correlation between contact order and long range order itself? Of course, they will be positively correlated, right? Because in both cases, we will see, let's say, higher value in case of long range contacts, whether it be sequence separation or whether a cutoff threshold above which values are considered as 1 or below which are considered as 0. So in both cases, we will see a higher value for the basically long range interactions, right? So long range interaction dominant structures, right? So in this case, the answer would have been on option C. So positive correlation between these two parameters. Now, earlier we talked about, like we saw one slide where the alpha dominant, beta dominant, etc. proteins where their folding rate were kind of put against the long range order and contact order and so on. Now, in the similar vein, if we ask a question, so we talked about the all alpha classes, which are mostly alpha helix dominant. We talked about the all beta classes, mostly beta sheet dominants. Talked about the alpha plus beta and alpha pi beta classes, which are mixed, right? So in which of these cases a relatively slow protein folding rate would be expected? Now we just talked about the inverse relationships between kind of in a broad way long range contacts and the protein folding rate, right? So that means that if we want to see a more slow folding rate, we have to look at proteins which are having higher long range contact formations, right? So in which of these cases basically a higher long range contact formation could be expected? So that is the question kind of asking in a roundabout way.
I guess all beta because there will be hydrogen bondings between the two sheets.
Yeah, exactly. So in this case, the more long range interaction dominant proteins will be the all beta classes, because if we just like take a look at the beta sheet dominant kind of structure, right? So the beta strands will be forming hydrogen bonds, right? So let's say if we have this strand, then it goes like this, then it goes like this, goes like this, right? So it will sort of have an antiparallel beta strand if we talk about it in this way, right? And then these interacting partners will be forming hydrogen bonds, right? So you imagine there will be a large sequence separation between these interacting partners, because in a different sort of strands residues are involved in the hydrogen bond formation. Conversely, if you look at a alpha helical structures, it is only n with n plus 4, right? So these are very closely situated residues that are forming hydrogen bonds in the alpha helical structures. With beta sheet, there will be a larger sequence separation between the hydrogen bond forming residues, because they are present at a different strand basically. So that means essentially the beta sheet dominant structures will have more long range contact formation, and they will at the same time show a slower protein folding rate. Okay, so that was the answer. So to take a quick look about the all alpha all beta proteins, we know that these are alpha helical dominant, beta sheet beta strand dominant, and then the all alpha versus all beta was that uh, in what way the alternate basically pattern was observed. So in the alpha by beta, we see just one beta strand after another alpha helix, another after beta strand, after another alpha helix, and so on. In the case of alpha plus beta proteins, we see a clumped uh, collective, basically alpha helices or beta strands in between, right? So that was the alpha by beta, also sorry, the alpha plus beta structure. And in these cases, of course, the folding rates will be different based on the contents of the alpha and beta. So here it is asking about a database that also Professor Gumiha mentioned in his slides, that is having data regarding the binding affinity of protein-protein complexes following mutations. So does anybody remember what this database was? So he introduced one database where following mutation, the binding affinity of protein-protein complexes was uh, detailed, right? So that particular database was called the proximate database. So you can take a look at this database maybe. So it is, I think, also basically maintained by the IIT Madras servers, right? So you can take a look at these databases and you can identify the mutation-induced affinity changes in protein-protein complexes, right? So these are just some of the types of questions that you can be expected to you can expect to get in your assignments and so on. So the concepts that Professor Gomiha always introduces in his classes and the numeric sort of examples that are coming into your assignments, like coming in your assignments. So these types of questions will also be present in the final exams as well, right? So you can take a look at these type of questions, try to solve some of those. Yeah, so in this particular class, I think we'll just wrap up this one. So again, thank you everyone for joining the class. Yeah, I think I see one hand in the chat. Yeah, please.
Sir, last time you had told me about your lab and all.
Yeah.
Sir, I actually visited your lab and I went through a few of your publications, but I was not able to download any of them because it asked
Oh.
for payment or institution access.
Yeah. So basically, since our institute has subscriptions to different journals, so we can access those. But of course, you are not able to outside any institute subscription. You have to pay. So I can share the publications from my lab with you.
Okay.
So you can share your email maybe, and I will send you the publication
Sir, actually,
articles.
I'll text you, sir. In half an hour, I'll text you through Google Chat or something.
Okay, sure.
Sir, last time I had told to take few papers, to consider few papers, and to redo those experiments, in silico experiments, and get the result. Try to get the result. Sir, I was not able to find like a particular paper like which. Sir, I was a little bit confused. Can you send few papers and guide me through it? Like,
Uh, sure, I could. Okay, so I can try to share my labs papers, some of my labs papers. Uh, some of the
okay,
simpler ones, for example, drug discovery, virtual screening, and so on. You can maybe take a look into those kind of papers and then try to see if you can replicate at least the virtual screening results or the adamant search results and so on, right?
okay, sir.
Yeah. So I didn't mean that you have to do the like entire process, but just bite-sized protocols that somebody has followed, and then try to replicate those kind of in a smaller scale, basically. So yeah, I'll try to send you the papers, and then you can maybe take a look into that as well. Okay.
Okay, sir.
Or alternatively, I can do one thing. So I think the PMRF portal has already shared the spreadsheet with you, right? Not PMRF, sorry.
Yes, sir.
The NPTEL platform. They have already
Yes,
shared the
sir.
spreadsheet with you, right? So I can in the shared Google Drive folder, I can upload some of the papers maybe, and you can take a look through that maybe.
Okay, sir.
Yeah. So in that shared spreadsheet, I'll upload some papers
Okay.
in a different folder, so you can take a look at that. Okay.
Okay, sir. Okay.
Yeah. Okay.
Thank you, sir.
Yeah. Sure. Okay. So again, thank you all for joining the class. Yeah. So please try to attend the classes of the other tiers as well if you find the timing convenient. And yeah, so we'll again meet in the next Friday's class. Okay. So thank you all.